you remember in the past? I mean, most people don't bring the handouts unless they post their lectures really late, like the night before. Then I tell them they have to bring them. So they stay up all night photocopying or whatever. But I try and, well, I do ask the lecturers to put up on Blackboard, put up the PowerPoint a lot earlier. Okay, I think we'll just go ahead. So some of you might have got the impression this was a 2-hour lecture every Tuesday and Thursday, because I think it actually said that on some of the course descriptions, which I have no idea why. This has always been a 90-minute lecture with no breaks. So hopefully that's good news. And my name's Jessica Tyler. I am the course director of this course. This course used to be called Eukaryotic Gene Expression, or EGE, and I changed it for this year just to more accurately reflect the content of the course. It's not just gene expression. You are going to hear a lot about gene expression, although over the years we've reduced that amount. You're going to hear about protein synthesis, you're going to hear about small RNAs, non-coding RNAs, you're going to hear about signaling, you're going to hear about DNA repair, replication, apoptosis, protein regulation through ubiquitination, and you're going to hear some bioinformatics, and also you're going to hear some basic cell biology. And so it's a lot more than gene expression. It's more accurately molecular biology of eukaryotic cells. There'll be no discussion of prokaryotic biology. Although, of course, research into prokaryotes was really important for getting us to where we are now with our current understanding of eukaryotes. Okay, so I'm the course director, and I'm going to try to come to all the good classes if I'm in town. But more importantly than me is this class has a TA. So I think we're quite unique in having a TA, at least for MD Anderson. And our TA is Avinash. Avinash, can you stand up, please? So Avinash is going to come to all the classes. He was specially chosen because he's a very strong, meaning a very good graduate student in our program. He did this course 2 years ago, and he's very interactive, very bright. He can help answer questions, whether it's to do with the science or to do with, you know, practical issues, like where's the exam, what kind of exams are going to be. Also, prior to exams, we post all the old exams that we can get our hands on, so that you have an idea of what to expect. This course is not aimed to be a memorization course. It's more aimed at teaching you the basics of fundamental eukaryotic processes that occur in the cell, with the aim of you understanding the concepts so that you can apply this knowledge to your own research. So a lot of the questions and the kind of questions that I try and encourage people to ask are questions based on, you know, a piece of data, and you have to analyze it. So we're really trying to facilitate your critical thinking so that you can, you know, understand a journal, literature, understand a research paper, ask how that might relate to what you're doing, and to help you design your own experiments to the best possible way that you can. And so what we've introduced new this year is we're going to, just going with that theme, is we're going to have journal discussion classes as well. Okay, so this is basically what I've told you already. We're not really going to stress disease in this class, but these are the basic processes that go wrong to cause human disease, particularly cancer. Okay, so the structure of the course is that there are 3 sections, so there's 3 exams. The idea is that you're not meant to, you know, memorize huge amounts of material and be tested on it 4 months later. So there's 3 exams, and all the classes are 90 minutes, and there'll be a review session prior to each exam. And the exams have been put multiple days after the last lecture, just so you have time to assimilate the material. I encourage you to come to review sessions because you can ask the lecturers anything you want, as far as, you know, you can ask them what's going to be on the exam. They won't tell you, but they'll give you a good indication of the kind of things that you need to know and that you don't need to know. Okay, again, the exams are to test your understanding of key concepts to try and help you better apply these concepts to your research. And the questions might also integrate different lectures. And as we go through the lectures, you'll see more and more that what you've learned earlier will be relevant to what you're learning on that current day. Okay, so we're going to have 4 paper discussion classes that have been put in at random, pretty much after a group of lectures. And how these will work is we'll divide the class up into groups. If we can get 4 rooms, we're going to divide you up into 4 rooms, and you'll be assigned either a postdoc or a faculty member who's an expert in that area. So for gene expression, you know, it'll probably be some of my postdocs or graduate students. And the idea is you'll read the paper ahead of time, and it will be to try and encourage you to, they'll ask you questions about the data and ask you maybe to point out which is the most important result in the gel. So, you know, first of all, it will all be very new to you, but after you've done this a few times, you'll get the idea. You won't be tested on these, but it will really help you in your exams, the more you participate in those. When I say participate, I mean speak, ask questions or answer questions. It's expected that you will go to these classes, just as it's expected that you'll come to all the classes in the classroom. Because if you don't, I mean, I'm not taking attendance, but you're only going to make your performance worse in the exam if you don't. And people have failed this course. Okay. Any questions? And before those paper discussions, we'll tell you what the rooms will be and what groups you're going to be in, and what room you'll be in. Okay, so what I've done today is I've condensed the 2 lectures that I've inherited, basically 2 subject areas, into 1 lecture. A lot of what I was teaching previously I actually thought was quite rudimentary and very simple. And so I just took out the really simple stuff and the stuff that's repetitive with what you're going to hear in future lectures. So I'm going to tell you about a few things. I'm going to tell you briefly about genes and genomes. I'm going to tell you about chromosomes and chromatin structure. In the next lecture, you're going to hear a lot more about the details of chromatin structure and how this regulates gene expression. Okay, so there's no text for this course. Some lecturers will give you assigned reading, and some lecturers will give you bonus points if you do something, but based on the assigned reading, perhaps. So these are texts that are useful in general for molecular biology. They're high quality, up-to-date texts. And that's another thing that I stress to all the lecturers, is that the idea of this, so you might have had many of these subjects before as undergrads, but the idea of this course is also to provide an update. And I tell them to update their lectures every year based on new literature. Okay, so even if it's familiar, hopefully it's a bit more up to date, rather than people that just teach from textbooks. And this is, you know, a pretty new textbook. And there's 2 copies of each of these books in the MD Anderson Library on reserve just for this class. I also have 1 copy of each of those. And so I'm on the 9th floor of this building, and so is Avinash. And so just come up if you want to borrow the books. Actually, I'll bring them to class on Tuesday, and someone can take them, and then when you're done with whatever you want to look at or photocopy, you bring it, and then someone else. You know, you don't have to do this reading. It's just if you're interested and want to learn more. Okay, so real basics. What is a gene? So a gene is the stretch of DNA that carries all the information to make either a protein or a RNA, like a functional RNA, a non-coding RNA. It's a unit of inheritance, and these are just different definitions, just mean same thing basically. But a gene includes the enhancer, the promoter, so these are the regulatory regions that are required for the accuracy of transcription, and you'll be hearing more about this with Shelley Barton's lectures on transcription. And of course it includes the open reading frame. And so through evolution, the number of genes has gone up as the complexity of an organism increases. And so this number, the number of human genes, is still a bit of a moving target, because all the time we're discovering non-coding RNAs that are encoded by genes. And so initially when the human genome was sequenced, they said there's 20,000 genes. But now our understanding of what a gene is becoming a bit broader, meaning basically with high throughput sequencing, people are discovering a lot of small RNAs that don't make a protein but are still expressed from a gene. So this number is probably going to increase. And so you can see here that even though, you know, we're going here from a single cell eukaryote, budding yeast, it has 6,000 genes, so it has, you know, about a fifth the number of genes as humans. But the general distribution of the functional classes of the genes is fairly similar through evolution. So a large increase in the number of genes has been due to duplication of genes and then specialization of those sequences so that they perform different new functions. So you've heard about gene families. So a lot of the increase in the number of these genes over a single eukaryote is due to gene families. Okay, so a genome consists of all the DNA in an organism. Usually we talk about, you know, the nuclear genome being the DNA in the chromosomes. So where else in a cell might you have DNA other than in our chromosomes? To speak?
Mitochondria.
Mitochondria, great. And you might also have a virus, a retrotransposon. But we're really going to talk about the eukaryotic genome, the thing that is, you know, constant between all of us. And so this genome obviously encodes genes, but there's a lot of extra DNA in the human genome that is called spacer DNA or repetitive DNA. There's also a lot of DNA that is transcribed, but is spliced out of the transcripts. These are introns. And this is kind of an old term that people use for DNA that they didn't understand what it did. It's basically similar to the spacer DNA. And now we know a lot of those DNAs actually encode non-coding RNAs. Okay, so just a generalization to remember is the size of the genome isn't proportional to the gene number. So there are organisms with really, really big genomes that don't have huge numbers of genes. So it's not a direct correlation. And these are just examples of model organisms that are used. Okay, so a lot of the reason why the genome size doesn't correlate to the number of genes is evolution. So some of you might think of budding yeast as quite a low organism because it is a single cell eukaryote. Budding yeast is actually extremely highly evolved. And during its evolution, it got rid of all the spacer DNA that it just didn't need. And so if you just look at a stretch of the yeast genome, each of these is an open reading frame. Yeast don't have many introns at all, and it really doesn't have much spacer DNA. Whereas by contrast, there's a similar length of the genome from humans, and there's things called pseudogenes. So these are genes that were made by a duplication event, and then a mutation occurred to get rid of the function of the gene, basically to stop it being transcribed. So there's a lot of pseudogenes in the human genome, and there's a lot of introns between the exons. There's a lot of extra DNA, so this is just showing ALU elements, which all have the same sequence, and they're scattered throughout the genome. And there's a lot of spacer DNA. So the size of the genome isn't always directly proportional to the number of genes. Okay, so the big surprise that came from the Human Genome Sequencing Project is that there are fewer genes than expected, because people originally thought, we're so complicated, we have to be made of, hundreds of thousands of genes. And so does anyone know some of the reasons why we don't have so many genes? I mean, what are other ways to make multiple proteins from one gene? Splicing, exactly. And so one thing that more larger eukaryotes have, so we say larger and smaller rather than higher and lower. Larger eukaryotes have is a lot of differential splicing. So that's combining different exons together, and you will have a lecture on that by Bill Mattox. Okay, so even though a very low percentage of the human genome encodes protein, a lot of the genome is transcribed into RNA. And this 28% may turn out to be higher because now there are these long non-coding RNAs that are being found. But basically, the main difference between this number is that around 27% of the genome is introns. So they get spliced out. They don't make a protein. And like I said, there's a lot more duplications of regions of the human genome. And this is thought to be important in allowing development of larger eukaryotes. And this parasitic DNA, such as ALU elements, makes up a large part of the genome. It's parasitic because it, in large part, originated from insertions into the genome, from either things that you don't need to know about, but retrotransposons and mobile elements. Repeating what we said. Well, actually, I didn't say this, but so it's changes in gene regulation and gene expression and alternative splicing that is thought to have played a really important role in evolution and adaptation to new environments, rather than gaining totally new protein functions. Okay, so that's the genome. But if every gene on the genome was expressed all the time, it would be a pure state of havoc. So it's really important to regulate gene expression. And if the genome was naked, meaning wasn't packaged together with proteins, the end result would be expression of all genes all the time. And so we're going to now talk about the epigenome. So the epigenome is a layer of information on top of the DNA sequence, which is the genome. And this layer of information is carried by chromatin structure and also DNA methylation. So we think of DNA methylation. So chromatin, I should define it. Chromatin is the complex of DNA and proteins that are bound to DNA. These include the histone proteins, but there's a lot more proteins that make up chromatin than just the histones. And so what this is showing is a chromosome. Actually, I got a better pointer. All right. That one died. Okay, so this is a chromosome at its most highly condensed state. Does anyone know what kind of chromosome that is? Or during what phase of the cell cycle?
Metaphase.
Metaphase, exactly. So people show chromosome like this because it's easier than drawing what actually a chromosome looks like in interphase, which is most of the cell cycle, which is just a great big huge mess. But during metaphase of the cell cycle, the chromosomes are extremely highly packaged. And this is so that there's it's actually two sister chromatids here, one here and one here. And it's so that they can separate accurately without the chromosomes breaking. And they can go to each cell. And you're going to hear more about mitosis also in lectures from Jill Schumacher. Okay, so there's two main ways that epigenetic information is carried in the chromatin. And the first of these is DNA methylation, which I'm going to tell you about. And the second is histone modification that I'm briefly going to introduce, and Dr. Jiabing Shi is going to tell you more next time. Okay, so just some definitions. I told you what chromatin is. A chromosome is the organized structure of DNA and protein that's found in the cells. So we have 23 pairs of chromosomes as humans. And each chromosome, well, this type of chromosome, a metaphase chromosome, has two molecules of identical DNA. That's because it's been through replication. But the DNA, if you add up the length of DNA in all 23 pairs of chromosomes, it comes to 2 meters per cell, which is really quite a long length if you think about how tiny a nucleus is. And there's, a huge amount, 50 billion kilometers of DNA in one human being. And so it's really important to organize this DNA and package it in a very organized way so that you can gain access to the right piece of DNA information at the right time. And that's the function that chromatin serves. So this is an overview of chromatin packing, and we're going to go through this step by step, where DNA rarely exists in a naked form like this in the cell. The DNA is naked for a really short time following DNA replication. But after that, it's very rapidly reassembled into this chromatin structure. So this is a depiction of a nucleosome, which is the basic unit of chromatin. This is a row of nucleosomes, and these are a row of nucleosomes coiled upon themselves. And then there are other levels of packing we're going to talk about. Okay, so this is a reenactment made from real structures of DNA and a real structure of the nucleosome. And they've added Brownian motion to make us think this is what actually might be happening. So these are the histones. They fly in, and actually it's a lot more complicated than this, but they result in the formation of this ordered structure called the nucleosome. And these things that are waving around, these blue things, they're the histone tails that we're going to be talking about, but are those major sites of histone modifications. And so now this is a solenoid, or the 30 nanometer fiber, and beyond this level it's all a bit of a speculation. But this is just one idea of how you might get a very ordered and regular packaging of the genome, which requires basically coiling upon coiling and coiling. And so you can think of it as a spring, if it was actually like this, that would have the potential to unspring and reveal DNA sequences at the time and the place when it was needed by the cell. Okay, so this is a real image of a dividing cell. You see the metaphase chromosomes. Well, now we're in telophase. Chromosomes going to each of the daughter cells. And then after this stage, the chromosomes decondense. So there's loads of these little animations that are really cool. There's ones for transcription, translation. They're kind of fun if you go to the website. Okay, so that would be great if we knew that was all true. Some of it's, from this level, is a bit of speculation, basically because the techniques don't exist currently to be able to determine really how the packaging occurs at these kind of levels. But we know We know what DNA looks like, we know what a nucleosome looks like. And the first level of packaging then is to this 10 nanometer fiber, which in this case, they've drawn it staggered a bit, so they've given it 11 nanometer diameter. This is also called beads on a string. I think I'm just looking to see what I've got here. I think later on I might have an EM image of that. I mean, it really does look like this. And then these, well, I guess this is going from highly condensed down to uncondensed. But I'm going the opposite way. This 10 nanometer fiber coils on itself to form this thing called the 30 nanometer fiber. And again, we're going to talk about these later stages, or at least what we think is going on. Okay, so this is the crystal structure of the nucleosome. The DNA, you can see, is strikingly bent around the outside of these histones. You all think of DNA as being a linear molecule, but the DNA is quite distorted to wrap around these histone proteins. And these histone proteins include two molecules each of histone H2A, H2B, H3, and H4. So there's 8 histone proteins, and there's 2 of each, of 4 different flavors. And the DNA wraps 1 3/4 times around the outside of those histones. So this is the nucleosome. And these histone proteins, especially histone H3 and H4, are so highly conserved through nature that, in fact, they're the most highly conserved proteins in all nature. So there are no proteins that are more similar from yeast to humans than the histone proteins. By similar, I mean the amino acid level. Okay, so I told you that after DNA replication, the DNA is transiently naked. So the black line here is the DNA. This is a nucleosome made up of the central core of histone H3H4 that's present in 2 molecules of each. And on the outside, there's a dimer of H2A, H2B, and another dimer of H2A, H2B, and the DNA going around the outside. Now, every time a cell replicates, all the histones are removed from the DNA, and then after replication, you now have these 2 daughter strands of DNA, and the histones are reassembled onto the chromatin. First of all, H3H4 goes down, because that's the center of the nucleosome, so that makes sense, that would go down first. And then these flanking H2A, H2B go down, and the DNA winds around them. And these histones are taken off and removed not only to allow, so the whole point of taking them off is to allow DNA replication to occur. Because during replication, you have to separate the strands of the DNA, and there's a really big machinery, the replication machinery, that has to move along the DNA, and the chromatin is just totally in the way. And so this also occurs during repair, where the histones are taken off to allow access by the machinery that does repair, and histones are returned to the DNA afterwards. And this also happens during transcription. Again, histones have to be turned off, removed from the DNA, to allow the transcription machinery to gain intimate access with the DNA sequence. And in the case of gene expression, this process of histones being removed from the DNA is regulated by transcriptional activators. So you're going to hear about these. These are sequence-specific DNA binding proteins that bind in the regulatory regions of the genes that they want to turn on, and they recruit other machinery that stimulates removal of histones from that very precise region of the genome around the gene. Okay, so the main function of chromatin then is to stop these processes occurring at the wrong time and the wrong place, because they basically block access to the machinery. So the chromatin allows all these processes to be highly regulated. Because if the chromatin wasn't there, first of all, the DNA wouldn't fit in the cell, which might be a minor problem, but also all these processes would occur all the time in the cell cycle, and it would just be a complete mess. Okay, so the core histones are very highly conserved at the the protein level, and they form dimers with each other. H2A and H2B form a dimer, H3 and H4 form a dimer. And they have this characteristic fold of a helix, helix, helix. And this is called the histone fold. It's also found in other proteins where they call it the histone fold. And so it's a very rigid structure that doesn't change. But what's not shown on here are the tails of the histones, which are these dotted regions. And why they're not shown is because they can't be seen in a structure. And that's because they're highly flexible, so they don't form crystals. And so the tails are at the N terminus of all the histones and at the C terminus of H2A. And these tails are the sites of the histone modifications in large part. Although I'm going to tell you there's also histone modifications within this structured region. And these histones are the most highly conserved proteins in nature. So what do you know about the charge of DNA? Is it positive, negative? Negative. And so to form this stable structure with histones then, that doesn't involve any covalent interactions, what do you think the histones would have to be? Positive. So the histones are highly basic. So what kind of amino acids?
Arginine, leucine, not leucine, but
lysine. Yeah.
Histone.
Exactly. So these histones are very enriched in lysines and arginines in particular. And this allows them to form hundreds, it's about 300 ionic interactions with the DNA. And it's these interactions within a nucleosome. There's about 300 interactions. And it's these interactions with the DNA that cause the DNA to bend around the outside of the histones. So, I mean, this is only 150 nucleotides. And it's causing it to bend twice, which is really quite striking. Okay, so here they've just drawn in the histone tails. You know, we have no idea what they look like, but we know that they head out past the DNA. Because in the crystal structure, you can see, you know, the part of the tail heading out through the DNA. So we think they're out here, and they're accessible to recruit proteins to the chromatin. Okay, so most of the histone DNA interactions are electrostatic based on the opposite charges of the histones and the DNA. And in red here, this is just showing where those positive amino acids are, and you can see they're laying right next to the DNA. So a lot of these interactions are with the phosphodiester backbone, not with the bases of the DNA. So unlike transcription factors that recognize specific bases and specific sequences, that is not the case with nucleosomes. They are not sequence specific in as far as recognizing bases. There is some degree of sequence specificity, but that's basically for DNA sequences that can be bent more easily. It's nothing to do with interactions with the bases. Okay, so this is just driving home this idea again that the DNA is wrapped around the outside. This is a surface of the histones that is accessible. If you rotate it, you can see the side view is mainly just the DNA. And this structure, the nucleosome, is present in all eukaryotes, and it's also present in some of the archaeobacteria. So it's a very old structure, and clearly it was a structure that, you know, was pretty good because through evolution it hasn't changed. So archaeobacteria are a bit different. They only have 4 histones molecules, not 8 in a nucleosome, but the basic structure is similar. Okay, so the DNA is quite bent around the outside, and so the nucleosomes don't bind to specific sequences, but they do like to bind to bendy DNA. And so even though the whole genome is coated in nucleosomes, and there's between each nucleosome there's about 20 to 70 base pairs, depending on the organism. That's called the linker DNA. So there's nucleosomes everywhere, but exactly where the nucleosome chooses to sit is determined by several things. One is how well the DNA can bend around the histones, and also the second thing is if there's a nucleosome right next there already, you're not going to get a nucleosome on top of that same DNA sequence. The histones are basic and they do repel each other a bit, so you won't get nucleosomes right next to each other. So there is always a linker DNA of some length, at least 20 base pairs. And if there's a sequence-specific binding protein, like a transcriptional activator, that will ensure that the nucleosome doesn't bind on that same sequence. So the locations of nucleosomes are determined by the proteins that already bind to the DNA or want to bind to the DNA. If there's a nucleosome there and a protein wants to bind, there are enzymes that will help the histones move along out of its way, or slide along the DNA to allow the transcription factor or the other sequence-specific binding protein to bind. And as I told you, the chromatin acts to limit accessibility to the DNA. And because of that, where the nucleosomes sit is quite important. So there's been a lot of interest in trying to figure out where exactly the nucleosomes sit on a piece of DNA. And a technique that you'll hear about through your careers requires on using the enzyme called micrococcal nuclease. So this is an enzyme that we add to nuclei. It's not present in humans. You know, we buy it from NEB, just like a restriction enzyme. And this enzyme is unusual in that it will only cut the DNA where it's accessible. So it cuts both strands of DNA in between the nucleosomes. And this is an enzyme that's often used in chromatin immunoprecipitation or chip analysis to fragment the chromatin. Right.
Where in between does it cut? Is it random?
It's not totally random, which it likes AT sequences. There is a preference. So the more enzyme you have, the more it will cut. So it depends on the kind of analysis you do. Sometimes people do a limited digestion, and then it will just cut at one point, just because it's accessible. And it will choose sequences with an A and a T. But if you give more of it, it will then cut again. And more you add, you can cut closer and closer to the edge of the nucleosome. So that is a problem with using micrococcal nuclease for chip analysis, is that it does have a slight sequence preference. So we've actually gone more to sonication, which is more random. But this is a method that, so it depends how accurately you want to know where the nucleosome is, because micrococcal will give you a slightly inaccurate result by, maybe 5 to 10 base pairs. But I'm not going to tell you about chip because chip will be gone through in detail in at least 2 of the other lectures. So don't worry, it will all become clear, but it's a very powerful technique for looking at anything that's going on chromatin, so repair, replication, transcription. Okay, so you basically cut up the whole genome with this enzyme, and there'll be little bits of DNA around 150 base pairs long that will not be digested by micrococcal nuclease. And then if the next step depends on your experimental question. If you wanted to ask, where are the nucleosomes on my favorite gene, then you would do a southern for your favorite gene. If you wanted to ask, where are the nucleosomes over the whole human genome, then you would go do high throughput sequencing of all the DNA and then map it onto the genome. You could also do PCR as well. And so using these kind of analyses, many labs now map nucleosomes. I mean, once it's been done in a wild type cell line or strain, there's no point doing it again. So people do it with their own favorite mutation or their condition to see, for example, when you give some growth factor to the cell, what happens to the nucleosome. So this is just the kind of data you get, where this would be from high throughput sequencing, where you'd map the number of reads against the DNA sequence. Then you get these peaks, and the peaks correspond to the center of the nucleosome. And so you can see positions on the genome where, places on the genome where the nucleosomes are highly positioned. And here, it seems like there's less nucleosomes, but they're still positioned. And there's also areas where there's no nucleosomes. Often at transcriptional start sites, there'll be depletion of nucleosomes. And this is to allow access of the transcription machinery to that area. Okay, so if a nucleosome is in the way, so let's say, this cell got irradiated with DNA damage and there was some double-strand break right here. And it's imperative that the cell repairs that double-strand break. In order to do that, the nucleosomes have to be moved out of the way. So if there's some kind of stimulus, the cell has mechanisms to move nucleosomes. And these mechanisms depend on 4 main activities. First, there are a set of proteins called ATP-dependent chromatin remodelers. So these use the energy of ATP, hydrolysis, to break those hundreds of histone DNA contacts. It's an extremely energy, I wouldn't say wasting, it uses a lot of energy to move a nucleosome. You need about 400 ATP molecules to be hydrolyzed to move one nucleosome to the adjacent position on the DNA. It's a huge amount of energy, but clearly the cell thinks it's important enough to do it, that it has these enzymes that do that. So they break histone DNA contacts, but they don't function on their own. They need other proteins that can bind to the histones to help them move along the DNA. These are called histone chaperones that help take the histones off the DNA, put them down somewhere else. There are enzymes that can modify histones on post-translational modifications on specific amino acids on the histones that can act to loosen or open up the chromatin structure to help with moving a nucleosome or totally taking the histones off the DNA. And there are also histone variants. So these are slightly different from the canonical H2A, H2B, H3H4. There are variants that are used because some of them form a looser nucleosome structure, and some of them form a tighter chromatin structure. So if it's a region of the genome that you want to be really highly inaccessible, like the inactive X chromosome of females, so female humans, we have 2 chromosomes, X chromosomes. Males have 1. So it's really important that only one of the female chromosomes, X chromosomes, is transcribed, because we need equal amounts of proteins that are made from the X chromosome, males and females. And so to do that, one of the female X chromosomes is made inactive. And how that happens is by packaging that X chromosome, only one of the 2, into a very inaccessible chromatin structure. And a main part of that process is using a histone variant called macro H2A that is very, it forms a nucleosome that's very solid, that cannot easily be moved. So that's an example of a histone variant. And there are other extremes where we're going to talk about histone variants that are used at regions of the genome that are transcriptionally active because they form looser nucleosomes that can be more readily moved out of the way, or the histones can be taken off. Okay, so ATP-dependent chromatin remodelers, they're a family of enzymes that are made of multiple subunits. They often have 5 to 10, sometimes 15 or 20 subunits. I'm not going to go through the details because it's just a concept that's important. But how these enzymes work is they have helicase-like subunits that hydrolyze the ATP. And as far as how we understand their function, they appear, so they're big enzyme complexes. And this is a cryo-EM image of one of these complexes, switch SNF, actually wraps around the nucleosome. And it kind of twists around the nucleosome, and as it does that, it breaks histone DNA contacts just for a short time at one place of the nucleosome, and then as it's twisting around, that region where the histone DNA contacts have been broken kind of moves around the nucleosome. So it can inchworm the DNA around the histones. Or if the histone DNA contacts have been broken, then another protein could grab the histones and take them off the DNA, like a histone chaperone. So they're fascinating enzymes. I don't actually think, well, there's a guy at Smithville that works on these, Snow Shen, and he's a real expert, but I don't think anyone here particularly looks at their mechanism of action. But the end result of their action is that this histone octamer, if it was on this piece of DNA, the ATP-dependent remodeling activity can make the histone octamer move from being there to being over there. And that will happen usually for a reason. The reason could be that another protein wanted to bind here, or other proteins wanted to gain access to the DNA during transcription or replication or repair. So there are ways to move the nucleosomes out of the way at the right time in the right place. Okay, so histone variants. Here's just a few flavors of some of the histone variants. We're going to talk briefly about some of these. There are variants of histone H3 and variants of histone H2A. There aren't really variants of H2B or H4. So H3 has a histone variant called H3.3, which is specifically enriched in areas of the genome that are transcriptionally active. And that's because the cell puts these down on transcriptionally active regions to allow the chromatin to be more open, to allow transcription to occur. We're going to talk about SEMP-A at the end. This is a centromere-specific histone variant. So it's the point where the mitotic spindle attaches to the chromosomes. And this SEMP-A histone variant forms the basis. Well, it basically tells the cell the position on which to assemble the kinetochore. So the kinetochore is the structure that attaches to the mitotic spindle. Okay, so H2A has several different variants. One of these, H2AX, is specifically used during the DNA damage response. About 10% of all H2A on the human genome is H2AX. It's randomly spread over the genome. But when there's a DNA break nearby H2AX, this gets phosphorylated by the DNA damage response, kinases like ATM and ATR, and that phosphorylation, it's called gamma H2AX. Any of you that have ever been in a repair lab might have heard of that. And this gamma H2AX then functions to recruit more proteins, like repair proteins, to that vicinity of the double-strand break. Okay, so H2AZ is a histone variant that often is assembled onto the genome together with H3.3, and together they form a very loose nucleosome that can be more readily removed during transcription. Macro H2A is this histone variant that is used to make very transcriptionally repressed chromatin. It blocks access to the DNA. For example, on X chromosome inactivation. And this is another histone variant that we. So, I mean, you don't need to know all these details. Again, the concept is that one way to change the chromatin structure, so influence the activity of the genome, is to incorporate histone variants that might either form a tighter chromatin structure or a looser chromatin structure. Okay, so I kind of pointed this out when we were looking at the nucleosome profiles, but active regions of the genome tend to lose nucleosomes. So repressed regions of the genome, that means transcriptional repression, is accompanied by having a high histone density over genes, basically. But active regions of the genomes have a lower histone density in promoters and open reading frames. So all of these studies are population-based studies. You know, we're looking at many cells at the same time. Again, we don't yet have techniques to look at where a nucleosome is in an individual cell. And so all we can say is that in the population, the occupancy is down. Whether that means, so there are less histones on that piece of DNA. Whether that means there are, 50% with lots or 50% with none, we don't really know. All we can say is in the population, there tends to be less histones over highly transcribed regions. And that's probably because instead what you've got sitting here is the transcription machinery, at least in some of the cells in the population. So I should say, whenever there's an arrow drawn like this, this is the transcription field's nomenclature for the start site of a gene transcription. So if that means transcription will go in that direction, and the initiation of transcription will occur here. If there's a cross through it, it means it's repressed. If the arrow is going, it means it's transcribed. Okay, so the idea of depleting histones is probably, it's probably just a consequence of transcriptional activity, that the polymerase and the machinery is going through the gene. So histone H3.3 is only different from what we call the canonical H3, the normal H3, by 4 amino acids. And so just to give you an idea of the length of these proteins, they're 135 amino acids long, approximately. So 4 differences is, 2%, 2 to 3%, I guess. So histone H3.3 has a few interesting characteristics in that it's expressed throughout the cell cycle, so it's expressed constitutively. Whereas the normal histone, or the canonical histone H3, which we also call H3.1, H3.2, these differ by 1 amino acid, but they, as far as we know, functionally they're very similar. H3.1 and H3.2 are only expressed during S phase. So why would you want to make histones during S phase? Someone from that side of the room.
For synthesis.
Synthesis of DNA, right? So during DNA synthesis, you go from having 2N, you basically double your amount of DNA. And it all has to be packaged into chromatin, so that's when you need to double your amount of histones. So there's a massive synthesis of histones during S phase, and then any extra histones have to be got rid of really rapidly. Actually, having a lot of these histones around will basically kill a cell, as we are discovering in our lab, which isn't good. But there's a lot of mechanisms that the cell has to ensure that you don't have too many histones. Probably because if you have lots of free histones, it's going to be hard to ever remove histones from the DNA. Because histones coming on and off the DNA is a constant equilibrium. And if you've got this massive pool of free histones, as soon as a histone comes off, it's going to be replaced from the free pool. So it's going to be hard for the cell ever to, make a region of DNA naked. And so the only histones that are around all the time, of the H3 flavor at least, is this H3.3. So if you're not in S phase, when a histone comes off, which occurs randomly, just because everything is in equilibrium, no interaction is permanent unless it's covalently linked. So I know we're all used to looking at structures and thinking about structures, so we think proteins, they look like that. All interactions are dynamic at some point. Even the nucleosome, which is an amazingly stable structure, it is dynamic. You get breathing of all interactions. So when a histone comes off outside of S phase, it's going to be replaced with H3.3. And the time when histones come off, usually outside of S phase, is during transcription, because a polymerase is barging through, giving a reason for that histone to come off. So once that histone comes off, it will be replaced by H3.3 and make a looser nucleosome so that the next polymerase can more easily push those histones off. So you don't have to use so much energy, presumably, to get rid of the nucleosome during the next round of transcription. Okay, so this H3.3 is deposited independently of replication, whereas H3.1 and H3.2 are only assembled onto the DNA during replication. And part of that is because that's the only time they're around, and part of that is because the way that H3.1 and H3.2 are assembled depends on the replication machinery interacting with the special histone chaperone that brings in H3.1 and H3.2. Okay, so consequently, we find H3.3 enriched in transcriptionally active genes. So H2AZ, or Z, depending where you're from, marks the transcription start sites. So what that means is when people have asked where on the genome do you find H2AZ, it correlates with being at the transcription start sites. So the state of the field of chromatin research, a lot of it is at this level of correlation right now. So people did ChIP-seq, which is chip analysis to map a protein, in this case H2AZ, over the whole genome, and they found that it tends to be on promoter regions. And when it's present with H3.3 at the same time, and so how you do this kind of analysis is you can look at both of them separately. You ask where is H2AZ, then you can ask in a separate experiment where is H3.3, and you find there's correlation. Or you can actually do a sequential analysis where you first of all ask where's H2AZ, then out of those that have H2AZ, do they also have H3.3? So that can be a sequential immunoprecipitation. And so they find that when there is H3.3 and H2AZ present, promoters together, these nucleosomes with H2AZ and H3.3 can be more readily removed from the DNA. And how they found that out was they isolated the chromatin and they washed it with salt, low amounts of salt. And so if you have salt around, that can break some ionic interactions. And they found that it then made these nucleosomes fall off the DNA, but not the canonical nucleosomes. So from that, you infer that a nucleosome with H2AZ and H3.3 is a looser nucleosome. That's kind of the state of the chromatin field as far as trying to figure out the functions of these histone variants. Okay, so we've talked about the 10 nanometer fiber, which basically is another depiction. The pieces of DNA between the nucleosomes, it's called the linker DNA. which I guess can be pretty broad in length. And the linker DNA is the site of where another histone called histone H1 binds. When histone H1 binds to the linker DNA, it basically causes the angle at which the DNA enters and exits the nucleosome. So here it's 180 degrees, or a straight line, right? So when H1 binds, this angle now goes to 90 degrees, causing this kind of zigzag pattern. And it's thought that the zigzagging of the chromatin is important for the next level of folding, which is coiling up of this zigzag structure. Now, this is what we call the solenoid or 30 nanometer structure. We don't exactly know how the nucleosomes sit in this structure. This is showing them coiling around each other, but there is still a matter of debate whether that's the case or not. It's still an area of active research. In this case, here's H kind of washed out, but H1 is sitting across where the DNA enters and exits the nucleosome.
So does it get wrapped up in there? Is that what they think? H1 is.
So it's outside of the wraps of DNA.
But in the 30 nanometer, it's still in there?
Oh yeah. Yeah, it's okay, it's not an enzyme, so it doesn't come do this and leave. It needs to be there. So the histones, you can think of them functioning, you know, in more of a stoichiometric manner. You know, for meaning then it's not a hit and run. So to form a highly packaged nucleosome like this, you have to have H1 there all the time. Yeah, so, you know, you can't see because of this color is the same as every other color, but the H1 will be on every nucleosome in there. That's a good question. Okay, so you're going to hear a lot about histone modifications, and I'm only going to tell you about 2 histone modifications. And that's because the 2 I'm going to tell you about are specific to chromatin packaging. The ones that Dr. Shee's going to tell you about are specific to transcriptional activation. So the 2 things are linked, but we're trying not to overlap. So he won't talk about the same ones. So one histone modification that's really quite unique and is important for going from this 10 nanometer fiber to this 30 nanometer fiber is acetylation of histone H4 on lysine 16. And that's because the evidence so far suggests that within the 30 nanometer fiber, this H4 tail right here binds to the adjacent nucleosome. So this one is sticking out and it binds to H2A and H2B on the surface of the adjacent nucleosome. And so by binding to the nucleosome next to it, it's locking it in to this structure that favors forming a higher order packaging. So as soon as acetylation of lysine 16 occurs, you lose the interaction between this nucleosome and this nucleosome. So this is an acetylation that is seen in highly active and open regions of the genome. When you lose this acetylation, the chromatin is more likely to fold into a higher order structure. So all these histone modifications and histone H1, ATP-dependent remodelers, none of them work on their own. They all work in concert. It's a combination of many things working together to change the chromatin structure. So this wouldn't be enough. I mean, it would be enough to stop that interaction if you acetylated it, but to change the chromatin structure, you're going to need additional activities. So try not to think of them as isolated activities. Okay, I knew I had this somewhere. So this is actually the 10 nanometer fiber, and you can see these balls of density. This is an EM image, and these are nucleosomes. And so this is partly why we don't know what the 30 nanometer fiber looks like exactly. We know its width, but how the nucleosomes sit in that 30 nanometer fiber, we don't yet comprehend. And, you know, people are trying to answer those kind of questions by gaining a structure of many nucleosomes. And it's believed that 30 nanometer fibers, or maybe a bigger fiber, attaches to something on the chromosome called the chromosome scaffold. So this is not protein, and it is not DNA. So if you digest a chromosome with nucleases or proteases, this scaffold still exists. So we don't exactly know what it is. It's a bit of a mystery, but it does appear to be, it is there. So this is, oh, sorry. So here they've removed